Let's take a look at the structure of some polynucleotides, so DNA and RNA. A DNA molecule is a double helix with two polynucleotide chains held together by hydrogen bonds between specific complementary base pairs. RNA, on the other hand, is made up of only one polynucleotide. So, the structure of DNA, so the bases are held together by hydrogen bonds. And there are 3 hydrogen bonds between guanine and cytosine and 2 hydrogen bonds between adenine and thymine. And the 2 polynucleotide chains or strands are anti-parallel, so they run in different directions in DNA. So I'll recap that. DNA is made up of 2 polynucleotide chains arranged in a double helix. The sugar and phosphate lie on the outside of the helix and form DNA's backbone. The nitrogenous bases are stacked on the inside of the helix, like a pair of staircase steps, and hydrogen bonds hold these base pairs together. RNA is a relatively short single polynucleotide chain. So there's only one chain, and it's comprised of ribonucleotides that are linked by phosphodiester bonds. There's specific base pairing, which means that only certain types of base pairs with other bases. So adenine in DNA binds with thymine, and guanine pairs with cytosine. This means that there's always an identical number of adenine and thymine bases and of guanine and cytosine bases in DNA. So this is called the base complementary rule, and this states that DNA strands are complementary to each other. If the sequence of one strand is AA, TT, CC, GG, the complementary strand would have the sequence TT, AA, CC, GG. Base pairing holds together the 2 DNA polynucleotide chains with hydrogen bonds, and the single polynucleotide chain is held together by phosphodiester bonds between the nucleotides. And they're anti-parallel. This means that the 2 polynucleotide strands run in opposite directions. The RNA-based sequence is complementary to the DNA's coding sequence from which it has been copied, except for that instead of adenine bonding with thymine, mRNA has a uracil, so uracil pairs with adenine. So the DNA bases that you'd find in DNA are adenine, thymine, cytosine and guanine, whereas in RNA you get adenine, uracil, cytosine and guanine. And that sums up polynucleotides for you.